My Dog is Broken by Zuhur Hassan and Milagros Santos. Signal transduction. What is signal transduction? Signal transduction is the process by which an extracellular signal can activate a membrane receptor and cause a cell to respond. For signal transduction to begin, a chemical signal binds to the extracellular part of the receptor and a conformational change occurs. The conformational change then changes part of the enzyme. Once this activation happens, a bunch of signals are amplified. This means that a small signal can lead to a large response. So in our case study, we looked at two different emails, and what seems to be going on is two friends having a conversation. One of them is a breeder and the other one is a doctor. She's a dog breeder, and she seems to be worried that her dog might have erectile dysfunction. And basically, for an erection to occur, blood has to rush through blood vessels. When blood vessels are made out of smooth muscle, and when these smooth muscles contract, less blood can enter through the blood vessels. When the smooth muscles are relaxed, more blood flow can go through the blood vessels. So pretty much as they're talking to each other, the doctor sends the breeder a picture of what exactly goes on in smooth muscle contraction. And right here is the picture, so let's talk a little bit about that. So in the beginning of any type of signal transduction, the first step is a signal has to bind to a receptor. In our case, for smooth muscle relaxation, the receptor is nitric oxide. Nitric oxide is a very small and nonpolar molecule, and it makes it easy for it to diffuse through the phospholipid bilayer. It does this through simple diffusion. This makes nitric oxide an intracellular receptor. So what happens is nitric oxide acts on an enzyme called guanylyl cyclase. Guanylyl cyclase is an enzyme-linked receptor. You can tell that it's enzyme because of the ACE at the end of cyclase. And what it does is it hydrolyzes phosphate. So what this means is that it removes two phosphates and forms a 3' to 5' ring structure. And it also forms GMP from the molecule GTP. GMP and ATP provide energy, and they act on protein kinase. They activate it. So protein kinase, it has two subunits that contain enzymatic activity by transferring a phosphate. That's the first one, catalytic. It contains an enzymatic activity by transferring a phosphate. The second subunit is called regulatory, and what that does is it binds cGMP to make it active. When PKG is activated, phosphoproteins lower the calcium in the blood. This lowering of the calcium leads to increased blood flow. And you can see in the picture, when an arrow becomes larger, that means that the cell signals are being amplified. So that means a small signal can trigger a larger response. And then after this, the guanylyl cyclase can be recycled all over again. So in the second email, the doctor had recommended two different treatments that were possible for her dog. Her first recommendation was the use of Viagra to treat the dog. What Viagra does is it works on phosphodiesterase to break down cGMP. And Viagra increases the erection by blocking the function of phosphodiesterase. In turn, the production of cGMP is slowed down. Therefore, the smooth muscles can relax and an erection can occur. The second treatment that she recommended was ginseng. Ginseng is known to increase the production of nitric oxide in the body, and this usually leads to an erection in males. So back to the picture here. Phosphodiesterase is an enzyme that breaks phosphodiester bonds. It removes cGMP once a signal molecule like nitric acid isn't present anymore, so then this turns off the function of the signal molecule. Viagra inhibits this so that it does not get turned off. This leads to increased blood flow and then the smooth muscle can relax. Ginseng, on the other hand, increases nitric oxide. So if ginseng was used for the dog, the amount of nitric oxide would be so high and more nitric oxide can enter the cell and trigger a cellular response. Also, ginseng would increase the blood flow by increasing the cell signal. So if you have more blood flow, that can go through the blood vessels and help aid in a reaction, in an erection. This is because of PKG. PKG activates proteins and lowers the calcium by being activated by cGMP. And then PKG affects other parts of the cell cascade. So PKG affects IP3, it affects P-lambin, and it affects the myosin light chain phosphatase. What IP3 is a secondary messenger. It's a membrane glycoprotein complex that targets calcium channels via facilitated diffusion. Calcium channels are found in the endoplasmic reticulum of cells. The IP3 closes the channels by phosphorylating the IP3 receptors, so when it gets activated, it releases calcium into the cytoplasm. When PKG is dysfunctional, too much calcium will be released, which would then raise calcium levels and decrease the blood flow. Phospholambin, or P-lambin, is a membrane protein that inhibits calcium pumps in the sarcoplasmic reticulum, and it does this via active transport, and it transfers calcium to the sarcoplasmic reticulum. When it gets turned off, it would leave the calcium in the cytoplasm. So if PKG is dysfunctional, it wouldn't be able to control the P-lambin and the calcium pumps would not work. So this would result in a buildup of calcium. Finally, the myosin light chain phosphatase is an enzyme that's important for regulating contraction in smooth muscle. When they contract, smooth muscle is relaxed. When it relaxes, smooth muscle contracts and becomes phosphorylated. This enzyme removes phosphates by phosphorylating the PKG. It is inhibited by the calcium pathways that maintain smooth muscle contraction. If PKG doesn't function correctly, the myosin light chains would contain too much calcium and the muscle contraction will not function properly. So if the ginseng and the Viagra don't work, then the most suspected molecule in this whole signal transduction would be the PKG, because it's almost the center of this whole thing. It controls a lot of different molecules, and if it doesn't work, then it will have an effect on the other molecules, and that wouldn't help the cell to continue its cellular response. So that's pretty much it. I hope you liked our presentation. Thanks for watching.